Hello, and welcome back to What is a Journal Club? I'm your host, as always, Mr. Johnny Hopkins. Today we'll be covering the folding of the hen lysozyme involves partially structured intermediates and multiple pathways, written by Radford, Dobson, and Evans. It was published, let's find this, 1992 in the volume 358 of Nature. So, let's figure out why we're even talking about some paper that was published a long time ago, about almost 30 years now. And so let's just get right on in and figure out why the folding of the hen lysozyme involves partially structured intermediates and multiple pathways. So, first of all, protein folding is a very, very complex and studied topic, especially by the Dill group at Stony Brook College. And in general, what they usually try and say is something called the funnel. There's a funnel that essentially you start at a denatured state over here, and then you go down along this this potential energy curve, this PMF, and go to a lower energy. So, but then you can find different metastable states, such as this little dip right here. It's a local minimum, but not the global minimum. So you can have these little intermediate states right here, right here, right here, right here. And those intermediates are kind of like, they're barriers. They have an energetic barrier that you cannot overcome going one way or the other, making them stable. But if it overcomes said barrier, it's able to go down and down and down, or in some cases it can go back up and make it become less stable. And so that's just a general idea of folding, is that you go from denatured, a wiggly boy, into a folded native state. So you got a wiggly boy, and then you say we have a helix with a beta sheet. And like that. But then if you have the intermediate state, this could actually in fact just have the alpha helix folded, and the rest of it is a wiggly boy. And so going on and on from there, you can go from denatured to an intermediate state to the native state. And the native state is typically just what happens in the cell, and the way you get to the native state typically is done in the endoplasmic reticulum of the cell with the help of different heat shock proteins and different chaperones and chaperonins that help make it work, as well as if you have bacteria, you got a trigger factor on the ribosomes that additionally help. So that's just, you kind of want to go down the funnel to try and find the lowest energy state, which is the native state. And so these guys, Radford et al., wanted to try and study this. And what they did to study this was they studied specifically hen lysozyme. Hen lysozyme looks like this guy, as seen in figure 2. It is not very easy to see here, but we've got a little bit of beta sheet here. You've got alpha helices going to B, then you've got some beta sheet, and then you got C and D helices, then you've got 310 helix as well. And yeah, that's a depiction of figure 2. So this is what the guy figures. So you've got 4 alpha helices, you've got 2 310 helices looped things, and then you got some beta sheets. And so this is a very good candidate to just study the movement from a denatured to a native state, because it's got multiple different structures, and see what goes on from going from here to there. So how did they do this? They used 2 different techniques primarily, well, 3 actually. So first of all, they used D2o Exchange Experiments with NMR. So effectively what happens is when you have D2O and a protein in solution, the D2O will exchange with the hydrogens that are freely available on the surface. So the D2O will attack those free hydrogens and come off. When I say D2O, I mean deuterium, which deuterium is just a hydrogen with a proton and a neutron in its nucleus, rather than just typically a proton in the nucleus. So that's what they usually say for proton occupancy rather than deuteron occupancy. And so this actually exchanges in its solution. So we can also have this H2O will attack the D2O as well in different cases. So when you have D2O though, you will usually, in NMR, you will quench the signal, you will decrease the amount of signal you get. And that's what they measured in these experiments as the D2O exchange over time. Further, they used circular dichroism. So this is an older technique and it's not very much as used in these days because it's rather insensitive, and you'll figure out pretty much why shortly. Is that essentially you have light that is circularly polarized and you shine it into your sample, and then eventually when it goes to the sample it will tilt a little bit and it will also like lengthen or contract depending on the thing, so you can get a different change in the angle and a different change in the eccentricity. And so it's usually measures just the delta epsilon of this eccentricity. And they also use something called stop flow, which I have covered previously, but in general it's just a way that you mix things together and then you take the measurement right when they mix, or 5 milliseconds after they mix. Just very, very quick measurements to get non-steady state kinetic information. So they applied all of this into this hen lysozyme trying to study the folding. So what did they find out? And what they happened to do, first of all, was the proton occupancy measurements. So they just essentially denatured the protein, and then they put it into a solution where they could nature it, and they measured the NMR signal at different points in time, and tried to determine the proton occupancy of different regions in the NMR signal. So with NMR, you can actually specify which individual proton you're wanting to look at, which individual signal of this guy you actually want to see. And so that's what they did. They tried to determine when it happens. And so when you look at all the helices pretty much that are the alpha helices, you can see just pretty much a single decay, pretty quick, and they all pretty much look superimposable upon each other. But then you go to the 310 ones, and then those guys have a very significant change in the rate, and as well as the beta sheet hat, it takes a lot longer to try to form and has a longer higher proton occupancy than these other two ones. So these processes are not one and the same time. Further, these fits that I'm showing the raw data because I am not that skilled. It's the whole thing with it is that so the alpha helices are able to just form a lot quicker because of the proton occupancy. That's just general. So essentially it seems to show that helices form faster than 310 helices, which form faster than beta sheets and beta strands. Because, well, as you can imagine, you have to coordinate these guys in 3D space. Whereas with the alpha helices, you can nucleate at any single point along the helix, and then it'll just kind of go from there. Whereas a beta sheet, you have to actually move these two little strands that might be thousands of residues apart, and you gotta move them together and get them close enough that they just kind of then nucleate and lock in together. So that is something that they found, is that there's not a single process. And what I was meant to say is that they actually did these fits with two, as a term of two exponentials, like a slow process and a fast process. And these slow and fast processes were able to then be, they're able to get a bunch of data about them and find out everything about every single residue, which, yeah, cool. Yeah, you can look at that in table 1, but I don't really feel like it's necessary to get into just, oh, this number means this and this number means that. Like, you just look at the graph and say, oh, it looks pretty much like that. So then we have figure 4 and 5. Figure 4 in particular actually is the refolding kinetics once again. So you go from unfolded to folded, denatured to native. And so what you can see is just the signal of this CD spectra for different lambdas. And you can see that this is very noisy data, but it does fit very well. Their fits do fit very well. And so it is very noisy. And going from there, they can actually look at the specific formation of different secondary structures in this. So what they actually found is that they're able to go from these guys and because they know different things about their instrumentation, they can actually extrapolate and figure out how much of the signal is actually alpha helix, beta helix, a secondary structure, or tertiary structure formation, just by looking at the signal from these wavelengths. And this is where it kind of gets a little bit tricky, is that you're fitting these equations to that to try to get these 4 different parameters, and it's not quite a perfect science and art. You can't really, it's a difference from instrument to instrument, and it's more of a qualitative measurement than you can get a quantitative measurement. You can get quantitative data, but you should always just make more a qualitative statement about it rather than a quantitative, and say statistically significant data, because this is fairly insensitive as shown by the high noise as well as fitting to different data. So going on from there, they're able to qualitatively show some different things. So you got just different formations, and they extrapolate down, and you can see at 0 seconds, you pretty much already have a bit of a secondary structure possible. That's what the 2 with the degree sign means. The 3 with the degree sign also means tertiary structure. So when you go to 11 milliseconds, you see a strong formation of even more secondary structure, with the alpha helix coming in about 50%, actually, maybe a little bit more. And then you have the beta and the tertiary structure forming even slower and slower, and then you got going up and up, and then they all kind of even out at 100%, where they're all pretty much good at 1,000 microseconds, one second. It takes 1 second for the protein to collapse in and form into the native structure instead of being just a regular wiggly boy. So, you're able to go from nothing to something by these measurements are able to, the experiments are able to recapitulate the data, showing that the alpha helix forms first and the beta helix forms second, tertiary structure forms the last, and which makes a lot of sense because, yeah, it makes sense. So we're able to see that different parts of this protein actually form at different points in time and take different amounts of time to fold. And so this is interesting, so it's not just one individual process, one sequence, but here's the next question that they have here, multiple pathways. Are there multiple pathways, or is it just one single sequential step? And although I'm not quite an expert on this, what they have here is they did the proton occupancy once more, but they altered the pH of the solution that it was in. And when they measured it, they saw effectively all the signal was the same across the board, independent of the millisecond, the refolding time scale of 11 milliseconds, 22 milliseconds, which they did not show the data, and 108 milliseconds. There was not a statistically significant difference between them. And so what actually they're able to conclude from this data is that there is not one single sequential motion. It just consistently moves and moves, but one protein can go down an independent kinetic path, and the other one can go down a different one. So what that essentially means is you can have one guy come over here and go down and down, and then be like, hey, I want to go back up, and go down and back up and down and then down. Or you can have another guy that comes from here, down, 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 back up, down, and down, and finally settle there. So they have different trajectories, different motions. They all have the same general trend, that the alpha helices form first, and the beta sheets form second, and tertiary structure forms last. But they all have different ways of going about in that direction. So you might actually have, say, helix B form first in one of the proteins, whereas helix D forms first in the other one, and then D helps C form, and then so on and so forth, and recapitulate the structure all down to the same structure in the end of the native one, but go in a very, very different, distinct kinetic route. So that is the entirety of the paper that I'd like to cover. And so one thing I'd just like to point out is some different quotes from the the conclusion-ish area. The 4-disulfide bridges persist in the denatured state and might have significant influence on the refolding mechanism, either through their isomerism in the denatured state or in intermediates, or through constraints that they place on the conformational freedom during refolding. So, one thing to remember here that I did not state earlier is that they make it into a wiggly boy, but it's not fully a wiggly boy. It's still connected with 4-disulfide bridges. So that could actually be an alteration that shows that this data is actually kind of not accurate when you think about it, because you're constraining the motion and you're getting it all back into the same native state because you've got these constraints in there. That said, it's probably unlikely, but you might be biasing actually the formation of alpha helices before beta sheets by just doing this. But that's highly unlikely. So realistically, the biggest difference here is simply that you probably have a longer time scale rather than anything else. And once again, disulfide bridges are also formed in the endoplasmic reticulum, the rough endoplasmic reticulum, because that's where everything with protein folding pretty much happens. And finally, these results have enabled us to develop a rather detailed picture of the folding pathway of hen lysozyme. CD and exchange labeling experiments have proved powerful and completely structural probes, revealing, for example, the very rapid formation of helices before specifically stabilized interactions are detectable. The refolding kinetics are complex, in particular because there is evidently more than one folding pathway. An important general feature not observed in other proteins studied by pulse labeling methods is the division of the protein into distinct folding domains, which, in most molecules at least, are stabilized asynchronously. So, as I have been showing you guys for a while, you're able to go from you can go from one denatured state and go down different kinetic paths to find the same one, and not all of the thing needs to form the exact same way, asynchronously, as they stated, and additionally, you can have different parts of it form independent of the other parts, and that can actually all form together. So that is the folding of the hen lysozyme involves partially structured intermediates and multiple pathways by Radford et al. I'm your host as always, Mr. John Hopkins. I'll see you next time. Thank you.